we can move on to our third and last talk. So Lauren Mak, can you share your screen?
Absolutely. Let's get that going. All right. How are you on visuals?
Yeah, I think we can see it okay.
Perfect.
So our last speaker is Lauren Mak, and she will talk about Ariadne, which is for barcoded linked reads deconvolution using the Bruijn graphs. Please.
Thank you. Thank you for having me here today. So my name is Lauren, and I'm studying with Dr. Iman Hajiya-Suliha at the Weill Cornell Medical College. And today I'm here to talk about Ariadne, which is a new algorithm we've come up with to deconvolve linked read clouds using only the reads themselves and a De Bruijn graph. So I'll be using the first few slides to go through metagenomic sequencing broadly, linked reads, and de novo assemblies. So if you're familiar with that, have fun on Instagram. I'll see you back in 5 minutes. So let's say we've written our grant, we've collected our sample, and we finally have a sequencing data set of our precious microbiome. We want to know either what it does, how it's changed, or both. But the first question we usually have to answer is who is there at a species level. So one way we could do so is by classifying reads using a reference database and tools like BLAST or Kraken-2. And reference databases are great, but they are informed by previous studies, which means that species in understudied environments are probably missing. So the set of taxa you do identify will be improperly biased to taxa that have already been characterized. So what are we going to do to find new species? That's which is what we want. So what we could do is shotgun sequencing. And the default technology for shotgun sequencing, which is short reads, doesn't really give you the sequencing breadth that long reads does, but long reads are expensive. So to balance the trade-off between those two aspects, we are exploring linked read technologies for shotgun metagenomic sequencing. Starting with our target microbiome, let's say we've extracted some long genomic fragments, and the goal is to make read clouds, which are sets of reads that carry the same universal molecular identifier, or UMI. So UMI is a barcode of between 12 to 18 base pairs tagged onto the 3' end of the read. So the example shown here is the 10x microfluidics method, where UMIs and long genomic fragments are encased in water droplets and co-amplified such that all reads from that same fragment carry the same UMI. And what I can do is, and I'm having problems with my pointer, but that's fine. Can you see my arrow or no? All right.
No, I can't see it.
Okay, all good. I'll just point verbally then. So if the median genome fragment generating this read cloud is about 20 kilobase pairs, and the average number of reads per read cloud are 6 to 270, you're probably getting a good approximation of nanopore reads. However, the converse, which is reads carrying the same UMI coming from the same genomic fragment, that statement is actually not true, because depending on our UMI distribution method, somewhere between 1 and 20 genomic fragments are associated with each UMI. As you can see in the graphic here, these blue reads and green reads, which are colored to indicate that they've come from different genomic fragments, they're still tagged with the same UMI because there aren't enough barcodes to go around. And the problem is that the assembled contigs from these mixed origin read clouds may contain genomic material from multiple species or large misassembly errors, not great for downstream analyses. So our lab previously published MINERVA to address this challenge, but because MINERVA relied on all-to-all k-mer comparisons between the actual sequencing reads themselves, it didn't really scale well to a data set of a realistic size. So enter Ariadne, which is our linked read deconvolution algorithm, which can be used to extract single species read clouds from a large linked read data set using information we'd get from a metagenomics assembler anyways. So Ariadne partitions the mixed read clouds into approximately single species sets, which are then used in de novo assembly to recover, hopefully, and what I will show, long contigs supported by reads from a single species. So before I jump straight into the algorithm, I'm going to go over a few concepts in the algorithm underlying metagenomics assembly. So we have a few things in mind before diving in. So I've got a sequence string here represented by this blue sequencing read. And what we do in panel A is decompose it into k-mers, which are used to populate the edges of a directed graph shown on the right here. So that's called a de Bruijn graph, and it uses edges to represent overlaps between strings. And after we've converted all the reads into k-mers, plug them into the graph, what we're trying to do is find a path through this graph that covers each edge exactly once. So for example, if we take that blue sequencing read on the left and we map it to the edges on the right, we can see that it forms a path through the graph. And this read contains a few repeats. So the path is back on itself a few times, covers some edges more than once. And this is actually fairly realistic because this is what happens at the end of de novo assembly when you generate these long contiguous contig paths through that graph. But the key point to keep in mind here is the idea of mapping reads back to the assembly graph. So on to what Ariadne actually does. So Ariadne takes as input a single read cloud and a preformed de Bruijn graph, which I use as one of the spades outputs. So this mixed origin read cloud up in panel A is comprised of 5 sequencing reads. The blue reads are from a single fragment, and a red read as from a different fragment. It just happens to have the same UMI. And Ariadne models each read as the center of a genomic fragment, therefore exploring the assembly graph both upstream and downstream in panel B should recover all of the edges that could potentially have originated from the same genomic fragment. So taking that one focal read, Ariadne maps it to the de Bruijn graph. And if we find the nearby edges surrounding that one focal edge highlighted, we'll be able to find also reads that from the same fragment that map to those edges. And conversely, reads mapping to other genomic fragments are likely to be excluded. So to do exactly that, Ariadne uses a Dijkstra algorithm to find the shortest paths between the end vertex of this edge up to a maximum search distance. The end vertex is just next to the C in the string in panel B. And all neighboring vertices up to like a maximum search distance is indicated by that black circle. So the set of edges included in the shortest path tree are the sequence space that represents the possible strings in the same genomic fragment. And as we can see, all the blue reads map to edges nearby, the red read maps to an edge much further away. And at the end, Ariadne partitions all the blue reads together into their own read cloud, and the red read is in its own little cloud to the right. So where does Ariadne fit in a de novo assembly workflow? Ariadne fits in immediately before de novo assembly, in the sense that it takes the original read clouds, generates the deconvolved read cloud subsets, which are then piped to an assembly algorithm to get improved contigs and scaffolds. And I use Cloud Spades as the de novo assembler throughout this entire process because it is aware of and can use the UMIs during contig generation. So as a performance test, I ran de novo assembly with or without Ariadne and a few mock metagenome datasets that were generated using a variety of linked read technologies. And I just want to stress that these datasets are generated from real mixtures of bacteria that were made in the wet lab. They went through actual lysis, DNA extraction, library prep, and sequencing procedures, because it was important to us to be able to validate Ariadne on datasets that were at least technically representative of a real metagenome. And as an added benchmark, I used a reference genome-based deconvolution method as an ideal best case scenario, which is where I mapped all reads to reference genomes that are known because they were mock microbiomes generated in the lab. I mapped them to reference genomes using Bowtie 2, and I deconvolved read clouds according to the species that the reads map to. So in the following slides, I'll be summarizing what Ariadne can do, as well as compare the performances of all three linked read technologies on data sets of, on microbiomes of a similar species complexities. So first, I wanted to look at the purities of the read clouds that we're looking at. The purity is the proportion of reads mapped to the most prevalent species in that cloud. So without deconvolution, which is shown in this red panel here, there's about 100% of the reads in mixed read clouds, as indicated by the bulk of these points. So that means if we were to randomly pull a read out of our FASTQ, it's probably in a read cloud that contains reads from multiple species. And as expected, there's some weak inverse correlations between the number of species and purity, as the average goes down from when the species complexity increases from 5 to 20, and also between cloud size and cloud purity, as indicated by the skew of the graphs. And the purity size graphs of the 10x and TEL-seq reads, which are panels 1, 3, and 4, have about the same shape, which is really interesting because TEL-seq and loop-seq both use transposase-based UMI distribution methods, whereas 10x uses its own microfluidics thing. So user beware, even if the technologies are on paper the same, you might not get the same distribution of read cloud properties. And after using Ariadne deconvolution on that original set of read clouds, over 75% of the reads in our data sets have been repartitioned into single origin read clouds, with the average purity going from about 45% before to about 90% now. And the 10x and TEL-seq graphs look pretty similar, but the loop-seq graph again stands out. And the purity actually decreases there from 93% to 88%. And I think the reason is because the largest original read clouds were split up incompletely. So for example, if you split up, say, like an 80% cloud into two smaller clouds that are 150% each, just by way of how we calculate the purity, the average has dropped. So the good thing about these Ariadne deconvolved clouds, is that they're unlikely to be artifacts, because I compared these to the clouds generated by the reference deconvolution, and the distribution is about the same, and there's about the same number of clouds. So in comparison to those original read clouds, Ariadne extracts a lot of single species information. And the next thing I wanted to look at was how much Ariadne deconvolved read clouds improved de novo assembled contigs. So the NA50 is X base pairs, where contigs larger than X contain at least half the bases of the assembly. And the longer and more contiguous the assembled contigs, the larger the NA50. So with the 5 species loop-seq and the 20 species TEL-seq datasets, there is a noticeable increase in the upper quartile of the NA50 range, so that's panels 2 and 4, and despite a negligible increase in the median NA50, which is indicated by the white line within each of the bars. So what that tells me is that even though most of the species may not benefit as much from read cloud deconvolution, there are still some cases within that single data set where having read cloud deconvolution will absolutely help contiguity. So while imperfect, it does seem to in some cases improve average contiguity more than doing nothing or the ideal reference-based deconvolution. So similar trends can be observed with in terms of looking at the alignment lengths between the assembled contigs and the known species reference genomes. And with the 20 species 10x data set, as you can see in panel 3, the median largest alignments between doing nothing and Ariadne, there's a big difference. And this is actually in contrast to the 20 species TEL-seq data set, where the converse is true. Ariadne deconvolution has generated, in terms of the upper quartile anyways, shorter alignments. So my takeaway from this slide is that while the results are slightly more mixed than with the NA50, there are at least some cases where Ariadne deconvolution generates slightly longer alignments. And I suspect that with a larger data set, as in more species in the original sample, there'll be larger gaps in performance between doing nothing and Ariadne. So improving contiguity and alignment lengths, that's great, but if we're also generating lots of misassembly errors in our context, that will absolutely confound downstream analyses, such as mag binning, or metagenome assembled genome binning as well. Misassembly errors come from aligning contigs to the reference sequences and seeing lots of large gaps come up. So if you had gene A and gene B adjacent on the reference genome, but they're 10 kilobase pairs apart in the contig, then that's a big misassembly error. So interestingly enough, the reference-based deconvolution contigs, so all the orange bars, had twice the misassembly rate of doing nothing in Ariadne, which is really strange to me at first, because that should be the best case, since all of the read clouds in that deconvolution are composed of reads from a single species. And my guess is that because there is more assembled genomic content on fewer contigs, you're just more likely to have large gaps come up because all of them are smashed together on that same contig. So despite having single origin read clouds, there might still be issues in the downstream product, which is why this is, and I think it's an interesting analysis. And in summary of this entire presentation, when we look at the different linked read technologies, the number of species do not change the shape of the size to purity distribution greatly, which reflects both inter-technology similarities, and it's a good sort of good thing to know that even if you apply this to a sample containing like hundreds of species, you're still going to look at about the same cloud size to purity distribution. And it's good for us because Ariadne is applicable to future iterations of linked read tech. And when you apply Ariadne to those read clouds, you get up to 37.5 times more single origin read clouds, which validates our sort of assembly graph-based radial search model of the original genomic fragments. And coming out the other end with Ariadne deconvolved contigs, we see moderate improvements in both contiguity and contig to reference alignment length, and a minimal increase only in the misassembly rate. And hopefully with future applications to real data sets, we're going to see more improvements. I'd like to thank the entire Hydrocillia lab, specifically Dima, David, and Varis for their extensive work on this project with me, Mason Lab for their incenting support in the wet lab, and our collaborators at USD and Loop Genomics, as well as funding support from NSERC. And with that, I would love to take questions. And this is a slide on where to find me and my stuff.
Thank you, Lauren. So currently, I don't see any questions in the Q&A, so maybe I will ask something while we wait others to type them in. So can you comment a bit on the runtime of this, of Ariadne? I mean, as compared to the, say, assembly that comes after it.
Absolutely. I'm going to stop this share and I'm going to share something slightly different because I actually have slides on that. Let's go to my apologies. I am bringing it up now. Share. So the runtime of Ariadne really depends on the maximum search distance you set to make the Dijkstra graph and to identify the edges surrounding the edge that the focal read mapped to. So we found that 5,000 base pairs was a good trade-off between deconvolution accuracy and runtime. So what I have in the top row here is the dataset name, which is our 5 species 10x dataset. And in the first row, I just listed the amount of time that assembly took and the amount of memory and number of threads. And if we use that as a comparison to using Ariadne with a 5,000 base pair search distance, it takes about 6 hours. So depending on your data set, it would take a different amount of time, and it would depend also on the assembly graph complexity. But overall, I think it would be reasonable to expect that using Ariadne on your full data set takes approximately, it's on the same order of time as you would use to de novo assemble your data set.
Thanks.
Yeah.
Now there are several questions, so maybe we'll take some of them live. So let's start with the first one. Isaac Coleman is asked, is this a question or comment? Okay, I'll just read it. Perhaps it is interesting to look at TNF distribution amongst reads with in-read cloud for reference genome read clouds versus real metagenomics read clouds. If the TNF frequencies are more similar for reference genome read clouds, perhaps this is the reason for more misassembly, more change of repeats. Were you able to parse the question?
Oh, okay. I think I understand. Yes. So I'm going to assume the question is, have I looked at the TNF frequencies in the read clouds themselves? I have not, and I didn't start with that because we had used a k-mer-based approach before, and that, well, yes, k-mers would obviously take longer than a TNF-based approach. We wanted to try something different, but that is something that I will probably go back to because I'm working on mag binning next, and I'm gonna have to start implementing those things anyways.
Okay, thank you. Okay, maybe there's also a comment by Ryan Chiki, but since this is more like a comment, then I will maybe just pass it on to the answered stack so that people can see it. And I think that's all that we have now. So thanks for the talk, and I would like to thank all the speakers of the session. And I think we do have a break now, right, Cynthia?
Yes, we have a short break now.